Good afternoon. I'm Owen Gorman with the U.S. Geological Survey. Today I will be speaking to you about barotrauma as a factor for survival in stock bloater. Before I begin, I wish to acknowledge my co-investigators Joshua Lyons, Tim Johnson, Aaron Fisk, Roger Gordon, and Kevin Loftus. Bloater were extirpated from Lake Ontario. The last specimen was caught in 1983. There have been ongoing efforts to restore bloater in Lake Ontario since 2012. Recent studies suggest that there may be high mortality of newly stocked bloater in Lake Ontario, and that will be the topic of my presentation today. Restoration of bloater in Lake Ontario began about 10 years ago with a wild stock propagation program using Lake Michigan bloater. Since then, rootstocks have been developed to ramp up production. Rootstocks are regularly augmented with wild stock propagation. The sequence begins with the collection of gametes from wild bloater in Lake Michigan during November-December, the spawning period. Bloater are captured in gill nets, and then the fish are stripped of gametes and eggs are fertilized. Those fertilized eggs are transferred to hatcheries for hatching, and hatched fry are reared, and the fry are raised up to fingerlings or larger life stages depending on the stocking plans. Fish are transported then from the hatchery to the stocking vessel and then are released at the surface of the lake at specific locations. Lake Ontario bloater stocking history began in 2012 with a modest stocking of a little over 1,200 fish and increased rapidly as the production capacity of hatcheries increased, reaching a peak of more than 300,000 fish stocked in 2016 and more than 200,000 stocked in 2017 and 2018, but then dropping to a little under 50,000 in 2019. And in 2020, the recent COVID year, the ability of hatcheries to produce fish and stock them was greatly reduced. And here we have a little over 16,000 fish stocked, the lowest number since the very first year of stocking. You will notice that over the years that there has been a shift from stocking fall fingerlings to stocking of yearlings and then also adult and subadult life stages. Overall, more than 1 million bloater have been stocked and more than 800,000 since 2016. As with any stocking program, there is assessment of success, and toward that end, extensive arrays of hydrophones have been established in the northeastern and western sections of the lake to detect acoustically tagged bloater. Also, stock bloater are vulnerable to capture in annual bottom trawl surveys along the south shore of the lake. So far, 53 tag detections were made outside the northeastern study arrays of the lake, and those detections occurred 4 to 179 days after stocking. There are also 10 bloater captured in the bottom trawl surveys over 2015 to 2019. A recent example of stock bloater captured in the 2019 bottom trawl survey is shown in the upper left. So the stocking strategy is to release juvenile fish, fall fingerlings, and fall yearlings, and during the daytime over deep water, 50 to 100 meters of depth, where they can descend to the relative safety of the lake bottom. It is assumed that this approach reduces predation mortality and is the same approach that has been used for decades when stocking yearling lake trout. Acoustics show that the fish swim downward to the lake bottom after release. Shown on the right is an echophotogram showing plots taken from the stocking vessel Ontario Explorer while bloater are released from the surface. What looks like a picture of rainfall are acoustic targets of individual bloater descending to the lake bottom, with some hesitation or pausing around the 50-meter depth, and then continuing on to the lake bottom. So the acoustic data confirms expectations that bloater descend to the relative safety of the lake bottom. So at this point, everything looks good. Now I will provide a quick overview of research led by Tim Johnson and Aaron Fisk to document the movement, ecology, and fate of stock bloater using acoustic technology. Their work was conducted in the northeast section of Lake Ontario, where an extensive array of hydrophones has been established to study movement of fish. In this slide, the location of the study area and acoustic detection arrays in the northeast section of the lake are shown to the right. On the lower left is Tim Johnson on board the RV Ontario Explorer holding a hydrophone, ready for deployment in an acoustic array, then deploying the hydrophone, and then a picture of the deployed hydrophone in Lake Ontario. In the center is Natalie Clenard in her capacity as fish surgeon, implanting an acoustic tag in a bloater. I will begin with the 2020 article in Freshwater Biology, Post-Stocking Movement and Survival of Hatchery-Reared Bloater Reintroduced into Lake Ontario. Natalie Clenard and collaborators conducted field research to understand the movement, ecology, and fate of stock bloater in Lake Ontario using acoustic arrays. Their work focused on an area in the northeast section of Lake Ontario where an extensive array of hydrophones was installed to track the movement of tagged fish. 74 adult bloater were implanted with acoustic tags and were released into Lake Ontario along with routine stockings of juvenile bloater. Survival of the 74 tag bloater was low. Only 47% survived the 12-day period of detection. Of the 59% that died, the majority died within 24 hours of release. Acoustic detection indicated that bloater swim rapidly to the bottom, disperse widely, and exhibit diel-vertical migration. An overview of the 159 bloater tagged between fall 2016 and fall 2017 shows that 66% of the fish died or were predated within 2 weeks of release. A common result is that more than half of the mortalities occur within 24 hours. But distinguishing between natural death and predation as the cause of mortality is not easy. I next turn to Clenard et al.'s 2019 article in the Journal of Fish Biology, Long-Term Retention of Acoustic Telemetry Transmitters in Temperate Predators Revealed by Predation Tags Implanted in Wild Prey Fish. A major objective of Clenard et al.'s research was to distinguish the causes of mortality in stock bloater. To that end, they used predation tags to distinguish natural death versus predation. Predation tags detect when fish are ingested and pass through the gut of the predator and then excreted. In this study, 47 adult bloater were implanted with predation tags and released in late 2018 into Lake Ontario. The timeline shows that all of the fish died within 7 days of stocking. 27 or 58% died within 24 hours. 25 or 53% of the fish were predated, and 22 or 47% died from unknown causes or disappeared, presumed dead. Tags ingested by predators allowed tracking of predator movements for up to 7 months. This research demonstrates that the majority of stock bloater are predated shortly after stocking, and that nearly half die from natural or unknown causes. These acoustic telemetry studies leave unanswered the question of what happened to these fish. What are we missing? Are stocked fish subject to some sort of physiological stress that leads to high mortality and increased vulnerability to predation? At this point, I want to summarize what we know about the fate of stock bloater. Hatchery-reared bloater have only experienced a maximum of 0.1 atmospheres of pressure in the hatchery. They are stocked during daytime over deep water, 50 to 100 meters depth. They swim rapidly to the lake bottom, achieving 5 to 10 atmospheres of pressure in as little as 4 to 7 minutes. Tag bloater show high predation, approximately 60%, and high short-term mortality, approximately 40%. More than half of the fish that are not predated die within 24 hours. So could these fish be suffering from compression barotrauma? Now, decompression barotrauma is widely recognized as a cause of death of fish. That is, when we bring fish up from depth too quickly. But compression barotrauma is undocumented in fish. There are simply no published articles on this subject to be found. So no one believes that this could be an issue, and finding funding has been challenging. In early 2020, the Great Lakes Fishery Commission provided $2,000 to fund travel to the Jordan River National Fish Hatchery to investigate the possibility that compression barotrauma could cause rapid morbidity and mortality in hatchery-reared bloater. But then the question comes, since this was in 2020, when are we going to be able to go to the hatchery to do this research? Well, we got our opportunity in November of 2020, and we gathered up our equipment and made a road trip to the Jordan River National Fish Hatchery. So what did we do? We have this hyperbaric apparatus for fish. We call it the HAF. This device was used to simulate the bloater diving to the lake bottom. We conducted 4 trials with adult bloater, and each trial took an entire day. The first 2 were rapid compression to 2 different depths, 50 or 70 meters depth. And so we got these fish down to depth in approximately 5 minutes or so to basically simulate what has been observed in the field. The second one was a slow 3-hour compression, taking 3 hours to reach the equivalent of 50 meters depth. And the fourth trial was the standard rapid compression to 50 meters of depth and holding these fish there for 3 hours. All fish were slowly decompressed for 3 hours or more. Each trial had a paired control, and more than 5 hours of video was recorded of fish behavior during these trials. The dead fish were dissected to inspect internal organs and to look for any signs of hemorrhaging. And the surviving fish were held for 30 days for observation. So here I present a montage of pictures of our work at the hatchery. We set up our portable laboratory next to the raceways inside one of the massive hatchery buildings, which we found tended to drop snowflakes on us during the morning hours. We took our fish from a next raceway where there are adult bloater for our use in these experiments. Each of the pressure tanks has a console with a live video feed, which you see here in the lower right. And there's also instruments at each one of these HAF units that monitors temperature, pressure, oxygen levels, which is logged on regular intervals. The fish are illuminated inside these tanks with infrared light. And of course, there's the control system, valves and levers galore to control the pressure within each of these vessels that we're doing our experiments with. So what did we find out? Bloater exhibited acute stress when compressed. Stress was evident at pressures as little as a half an atmosphere. That's the equivalent of 5 meters of depth. And we could see this within minutes of the very first trial. Moderate stress is indicated by increased respiration and activity, swimming speed, etc. Acute stress is indicated by disequilibrium and lying motionless on the bottom, or on their side, or upside down. 44% of the fish reached this state in the compression trials. Decompression alleviated the symptoms of stress caused by compression. After the trials, of the fish that were compressed, 19% of the fish died within 48 hours, and those mortalities increased to 25% within 7 days. 6 of 7 of those mortalities had ruptured swim bladders, and that provides evidence of compression barotrauma. No control fish died. So in conclusion, we found evidence that compression barotrauma causes morbidity and mortality in hatchery-reared bloater. So I'm going to just show you a short video clip of what these fish look like inside these pressure vessels. On the left are the fish under compression under the equivalent of 50 meters of depth. In the control are fish that are at sea level, essentially no pressure at all. And you will notice a rather big difference in their behavior. So what you see on the compression side, you see a lot of particles being stirred up, flotsam in the water, caused by a lot of the fish movement. There's two fish lying on the bottom. And whereas you look on the control side, these fish are just gently gliding back and forth through the screen in view. So the implications of our research thus far is that compression barotrauma may affect fish stocked over deep water. And this has implications for current stocking practices. They may need to change. And this is all pertinent because there is interest in restoring kai, a deepwater cisco, into Lake Ontario. We still have a lot of work to do on this project, a lot of fish to dissect and look at, analyze a lot of video footage, etc., to better understand what we have at hand right now. But we also want to be able to look at the effects of compression barotrauma on sizes of bloater that are typically stocked in Lake Ontario. That would be fingerlings and yearlings. To that end, we brought 500 baby bloater back to our station in Ashland, Wisconsin. So to support this, we are looking for additional funding, and there is interest in expanding the scope of the study to include cisco and lake trout. And all of this may provide a guidance for improved stocking methods to reduce the likelihood of barotrauma in efforts to restore bloater and other species in Lake Ontario. So this research is really in its starting phases, and it will be continued. So in closing, I wish to acknowledge the collaborators in this study and those that have made contributions, and to the Great Lakes Fishery Commission and U.S. Geological Survey for their funding of this research. Thank you for listening.